All right. So my name is Tracy, Tran, and I want to start first by thanking the organizers for giving me the opportunity to talk about the new research in our lab. So I'm from the McMahon lab, and for those who know the history of the lab, we have a long-standing interest in kidney development, especially studying the interaction between different progenitor population and how they give rise to nephron formation. So my interest fits into the lab interest that I want to study these interactions, but in the context of human kidney development. And we obviously don't have that many models out there to study human kidney development, but the newly emerged in vitro nephrogenesis system may help with that. So that's the story that I'm going to tell you today. That first, I'm going to tell you about the characterization work that we've been doing in the lab and how we use that information to evaluate the in vitro nephrogenesis system. And the goal for today's talk is to get your attention on kidney regenerative research. We're a little bit behind compared to other fields in the organoid field, but I hope to get your attention on that. So to give you a little bit background about kidney development, the nephron, which is the functional unit of the kidney, forms as a result of complex interactions between the different cell types in the nephrogenic niche. So I'm showing you here an illustration of a mouse embryonic kidney, and for each of these dumbbell structure, this is a nephrogenic niche, and we have a staining on a sagittal section of the mouse nephrogenic niche. The nephrogenic niche is composed of the metanephric mesenchyme population interacting with the ureteric bud tip. The metanephric mesenchyme is made up of the interstitial progenitor cells that are marked in green here, and the nephron progenitor cells, which are Sixl positive. The nephron progenitor cells interact with the interstitial progenitor cells and the ureteric bud tip and become induced. So these induced cells will give rise to the aggregates underneath the ureteric bud tip. And these induced cells will further mature to become renal vesicle, now polarized into distal and proximal segment. And the renal vesicle will further mature to become S-shaped body, now segmented into 3 segments and get connected into the ureteric bud. So together with the ureteric epithelium, the S-shaped body will give rise to different compartments of the nephrons. That is a really complex network. So recently we have a few protocols that were published that they aim to make kidney organoids from pluripotent stem cells, but a few questions were arise from these work. That first of all, do the kidneys to the cell types that are made in the organoid, are they actually kidney cell types? And the second question is that the formation of these kidney organoids actually reflect normal human kidney development. So we realized that the field doesn't have a standard to evaluate the kidney organoid in vitro, and we decided to go back to do characterization on the human development and see if we can establish a standard to evaluate these work. So that's fit into the outline of my talk today. And to characterize the human kidney, we use the mouse as the basis and compare human nephrogenesis to the mouse and ask what are their similarities and differences. And to do that, we integrate different technology that we use first 3D images to get an overview of their morphology. For 2D images, we get a cellular anatomy view that we go into very detail of what are expressed in these cell types. And to understand the human kidney development at molecular level, we employ different sequencing technology like RNA-seq, ChIP-seq, and ATAC-seq to create the profile of these cells at the molecular level. So I'm showing you here the top view of a week 13 human nephrogenic niche that you can see very clearly here. Nephrogenetic cells are forming a really tight structure around the ureteric epithelium. And from these 3D imaging, we were able to do a lot of quantification to study the dynamic of human kidney development. One example that we have done is that we quantified the nephron progenitor cell endowment over time and compared that to the mouse, and we found that very similar to the mouse that as nephrogenesis proceeds in human, we will see a reduction in the nephron progenitor cell endowment. We were also able to look into the brain gene dynamic that at the early stage of nephrogenesis, brain gene would be more active at around week 11 compared to the later stage that we were able to look at. That here you see the branch points were really deep into the tissue. That means nephrogenesis has happened without branching. So that was the 3D view. So how is the 2D view? In the mouse, so this is the sagittal section again that we see the cap mesenchyme here. It's a nephron progenitor cell population interact with the ureteric epithelium. And these are the really early renal vesicle developing nephron structure that we see in the mouse as marked by the induction marker, jacket 1. So in the human, it has very similar organization, but the structure are much larger, and you see the lumens much clearer. But also in the human, we see something that were not noticed in the mouse before, that this is the developing nephrons here, and we cells recruited from the cap mesenchyme into this developing nephron structure that hasn't described in the mouse before, that we think this is a novel mechanism of self-recruitment into the developing nephron. Going to another stage of nephron development, this is the later stage, the S-shaped body. So again, very similar to the mouse and the human, S-shaped body are segmented into 3 different segments, distal, medial, and proximal. But again, the human structure are much larger, and lumens is are more obvious in the human case. So the 2D view is very beautiful already. But we're not very satisfied with that, so we're going up to another level. So we want to obtain a 3D view of our developing nephron. And I think this is the first time that we see very clearly that the nephrogenic niche in the human is more complicated than we think. This is a pre-tubular aggregate and clearly it's not the cells are streaming not only from one nephron progenitor population, but 2 nephron progenitor population. And the S-shaped body here is not just an S-shaped body like we see on the 2D, but it's actually more complicated than that. It's more convoluted, especially on the proximal segment that are giving rise to the glomerulus and the kidney. And we also see nephron arcading here that I'm not going to go into detail today. So morphologically, they're very similar. How about at the molecular level? So we were able to identify some differences across the species. One of the standout difference between human and mouse is the expression of Sixl gene. So Sixl1 in the mouse, together with Sixl2, would mark the very early metanephric mesenchyme population. These 2 genes are very important for establishment and maintaining the metanephric mesenchyme population. But only Sixl2 would persist until almost the end of nephrogenesis. Sixl1 would turn off at about 11.5, even though it's very important, it's required for the establishment of this early metanephric mesenchyme population. In the human, however, we see Sixl1 persist until as late as week 23 of gestation. So what does that mean? So given the role of Sixl1 in nephrogenesis in the mouse, we think that the presence of Sixl1 in these nephron progenitor cell population in the human may promote the self-renewal period of these nephrogenetic cells in the human. That's how we sustain the really long period of nephrogenesis in the human, which is about 30 weeks compared to a mouse, which happened only in 2 weeks. And we're going further to explore the regulatory network of these two genes, Sixl and Sixl, when we found that in the human, only in the human, that Sixl is a target of Sixl. And this work has been published that we are going to further the interactions between the regulatory network of Sixl and Sixl. So the gypsic work is just one example. We have another work in the lab that employ Mars technology to isolate out different cell types in the human kidney, because obviously we don't have human GFP positive cells from the human samples. So with this technology, we were able to isolate out the Sixl positive population by performing Sixl transcription factor staining, and then isolate these two out and do RNA-seq on these. And these are just some highlight of this human unique genes from the Nephron populations. And this is just one cell population that we are isolating out, and we aiming to isolate other populations in the human. So how are we using this work to evaluate the in vitro nephrogenesis? So I told you earlier that we have a couple of protocols out there to make kidney organoid, but as a development lab, we are interested, in order to use this system for human kidney development study, we have to be certain about two things. So first of all, the formation of a kidney organoid has to recapitulate the formation of normal human kidney. And second, so the cell types that are made in the kidney organoid have to be kidney cells. So we employ that protocol in our lab by using human ES cells instead of iPS cells. And our couple first attempts are very potential, that we see cells that are positive for WT1, LTL, and ECAT, forming very distinct structure in our organoid. That WT1 positive cell would organize around the periphery of the organoid, and LTL, ECAT, and WT1 cells would form a network inside of the organoid, and WT1 and the major nephron would mark the glomerulus, cell in the glomerulus. LTL would be the proximal tubule, and ECAT would mark the collecting duct. And when we look into the zoom-in picture of the organoids, so these WT1, LTL, and ECAT positive cells form connecting structure that they form in the order very similar to the order in the real nephrons. But of course, morphologically, they don't look very similar to the real kidney at all. So we have to characterize these structures very carefully. And of all the things that we look into, we have strong expressions of genes that are markers of the podocyte population, So podocytes are the cells that wrap around the blood vessel in the renal corpuscle. And they are the cells that form the very first filtration when blood enters the nephrons. And these cells are very susceptible to kidney injury because this is the first receptor received site when blood enters the nephrons. So in order to isolate these cells out from the kidney organoid, we have the MAFB knock-in cell line. That MAFB would mark the podocyte population in the real kidney. And at the end of the protocol, it's very happy that we have GFP positive cells around day 20. And by immunostaining, we were able to verify that the GFP positive cells are also MAFB positive cells, which we want the GFP to report, and Nephron positive. And look into the more mature structure, the GFP positive cells are also clustered in these cells are clustered into a small group, and they are surrounded by the epithelium structure that are keratin A positive, which is quite similar to the normal organization of a normal glomerulus. And we sorted out these cells, and at this point, we have the RNA-seq profile of these, and compared to the GFP negative positive, we have enrichment of genes that are normally expressed in the podocyte population in the human. So there remains a lot of open questions from the in vitro nephrogenesis work. So still, going back to the question, does in vitro nephrogenesis recapitulate human kidney development? Even though we have the Nephron progenitor cells marker reporter line, we haven't been able to isolate out Nephron progenitor-like cells from the kidney organoid. Maybe we have missed the window, the very short window that they express, that they show up in their kidney organoid. What cell types are formed in kidney organoid? So I've just showed you one example of the MAFB EGFP cell line. We also have other cell lines to isolate proximal tubule, and that would be a continuing work to verify this identity. Do Nephron cell types exhibit mature cells transcription profiles? So one concern that we have from the MAFB EGFP positive cells that we isolated out is that some of the expression levels are not as high as the glomerulus profile that we have in hand. And that's about the problem of maturation. And the next step would be vascularize the organoid to make them mature. So that's remain a lot of work for us to do. I want to end this talk by embrace you that the kidney nephrons is a very complex network that you have to have the right cell type interacting with each other, and then those cell types would form tubules that connect to each other. And with that, I want to end my talk with thanking my mentor, Dr. Andrew McMahon, for all of his support. And these are the people that who were directly and indirectly involved in this work. And with that, I want to take questions from you. Thank you.
Very beautiful pictures, impressive. So, I have a question.
Congratulations to that work, and it almost looks as if the kidney is almost as complex as a brain. So,
Thank you.
I had a question regarding the podocytes with the expression of podocin and the nephrines. I mean, these are involved in formation of the slit diaphragm. Have you seen any indication that they actually form these interdigitating processes and that they form sort of a slit diaphragm?
So that's a great question. So one thing that we're missing from our organoid is the vascular system. And in order to form the slit diaphragm, you have to have the podocyte wrapping around the vascular system. So that we haven't had. But from the published work, it seems like it's had the potential to form that slit diaphragm. But definitely not as fabricated as, as complicated as the slit diaphragm that you have in the real human kidney. Thank you.
I have a follow-up question. So how do you guys are thinking about vascularization? Is it going to be something that you're mixing cells or you're trying to create a structure to vascularize?
Yeah, so one very important thing that we have to take into consideration is that what do you want to use these organoids for? So but just for the purpose of drug screening, we have to use something that is totally in vitro, and maybe HUVEC cells and mesenchymal stem cell mixed in there would help with that. But if we want to use like a real vascular system, then we have to put into the mouse kidney capsule for the full vascularization. So it depends on the purpose. If we really want to make a really beautiful kidney organoid, then maybe surgery and put them into kidney capsule would be the way to go.
Have you guys tried to do that with the human cells? Do the mouse vascularization will be attracted to the human cells?
Yes, so people have done that, at least with their cells that are isolated out from the mouse kidney or the human kidney, and then they put back into the kidney capsule, yes.
I see. Okay. All right. So if no other questions, let's thank Tracy again.
Thank you.
Thank you.